Cardiovascular disease is a major cause of death in Western societies. But what do we know about the role that genetics and lifestyle play in cardiovascular risk? And how can we use our understanding of lipoprotein metabolism to understand genetic risk of cardiovascular disease? In this video, we're going to talk about the relative roles of lifestyle versus genetics in predicting cardiovascular risk. We're going to use our knowledge of lipoprotein metabolism to understand why specific genes are associated with both changes in lipoprotein particle numbers and cardiovascular disease risk. We're then going to integrate that with how we predict cardiovascular disease risk and what are the clinical risk factors that we can use to measure cardiovascular risk, which are the most effective for understanding whether or not somebody might be likely to have a heart attack. Over the last 20 years or so, we've learned quite a lot about the genetic risk of cardiovascular disease. For example, shown here is the lipoprotein lipase system. LPL is important for delivering lipids out of chylomicrons and VLDL particles and into circulation. Inefficient lipoprotein lipase activity will result in lipids maintaining in our blood. This in turn will elevate cardiovascular disease risk. Monogenic syndromic variants of several of these enzymes have been associated with changes in lipoprotein metabolism and cardiovascular disease risk. For example, both ApoC3 and ANGPTL4 are negative regulators of lipoprotein lipase. That means if you have a loss of function in either of those two proteins, lipoprotein lipase will be more active. If LPL is more active, you are more likely to get lipids out of your blood and into your peripheral tissues. In turn, that reduces cardiovascular disease risk. On the contrary, ApoA5 and lipoprotein lipase itself are both positive regulators of LPL metabolism. That means if you have a loss of function of LPL or ApoA5, LPL is less active. That means lipids are more likely to stay in the blood. And that means they are more likely to cause cardiovascular disease. And this is exactly what we find when we examine individuals who have major loss of function variants in ApoA5 or LPL. The reason for this is through the buildup of LDL particles. LDL is generated by the breakdown of VLDL. And if LDL stays in our blood over time, it can penetrate into the endothelial wall of a cell on a blood vessel. These LDL particles can clump together, forming an atherosclerotic lesion. In addition to inflammation, that can result in an atherosclerotic plaque. Over time, that plaque could become disengaged, forming a thrombosis. This blood clot can then go and occlude other blood vessels, in your coronary artery, causing a heart attack, or in your brain, causing a stroke. A lot of what we understand about cardiovascular disease risk comes down to understanding what elevates LDL numbers in our blood. There have been about 50 GWAS associations of individual genetic loci with cardiovascular disease risk. We can take those 50 GWAS associations and generate a polygenic risk score across all of those alleles. Based on this, what we've learned is that cardiovascular disease is 50 to 60% heritable, mostly due to changes in lipoprotein metabolism. This study here is a 3x3 summary of cardiovascular disease risk. Individuals are broken down into 9 groups. Those with low, intermediate, or high genetic risk based on their polygenic risk scores. And then within each group, people who had favorable, intermediate, or unfavorable lifestyle. This is defined by their smoking status, obesity, physical activity, and what was termed a healthy diet. What they found is within each group, a more unfavorable lifestyle is associated with higher genetic risk of cardiovascular disease. If you look at the high genetic risk, high unfavorable lifestyle group compared to the low genetic risk, most favorable lifestyle group, there is a very pronounced lifestyle effect, going from about a 2-fold increase to about a 3.5-fold increase. Another thing I wanted you to take from this, though, is if you look at the high genetic risk but most favorable lifestyle group, they have about the same risk of cardiovascular disease as the low genetic risk, most unfavorable lifestyle group. What this tells us is that lifestyle and genetics both contribute about equally to each other. Within each group, lifestyle modification is an important way by which we can reduce the risk of cardiovascular disease. On the other hand, genetic risk can tell us, even as somebody's been born, what their overall genetic risk might be, so we can target lifestyle changes to those who are at the highest overall genetic risk. Shown here is the association of LDL cholesterol levels to cardiovascular disease risk. Shown in blue is Mendelian randomization studies. These are studies where people have genetically predicted elevated or reduced LDL cholesterol levels. They're generally born with variants that cause them to high LDL-C throughout their lifespan. For example, somebody who has an LPL loss of function variant. As you look down this line, the more a variant affects your LDL-C, the more it affects your cardiovascular disease risk. In the next line down are prospective cohort studies. This might be looking at adults and then looking at their longitudinal likelihood of having cardiovascular disease, then associating that at what their LDL-C levels were during adulthood. Again, having higher LDL-C levels during adulthood is associated with more cardiovascular disease risk, and having low LDL-C is associated with having lower cardiovascular disease risk. Finally, the last group is randomized clinical trials with LDL-lowering medications. The extent to which that medication lowers your LDL is linearly associated with the extent to which it reduces your cardiovascular disease risk. Now, if we compare across these groups, what this is telling us is lifetime exposure to high LDL-C is very important. The people in the Mendelian randomization studies have had elevated LDL-C for their entire lives, so they've had a lifetime exposure of high LDL. On the other hand, individuals who are in the RCTs, their LDL was only changed during the study. So therefore, the impact of that reduced LDL-C is less. Prospective studies fall somewhere in the middle. Importantly, understanding cardiovascular risk involves not only just understanding changes in LDL-C, but understanding how long elevated LDL-C has occurred. Because cardiovascular disease, unlike hypertension or diabetes, isn't a continuous event, it's a singular event, we rely on surrogate blood measures to identify prospectively who is at elevated risk of cardiovascular disease. Some common measures are total cholesterol, LDL cholesterol, and HDL cholesterol. This is related to, but actually distinct to, the number of particles of each of LDL or HDL. Because each LDL has one and only one ApoB100 molecule, and every HDL has one ApoA1 molecule, we can measure the concentration of ApoB100 or ApoA1 to determine the number of particles in the blood. Again, this is related to the amount of cholesterol in that fraction, but it's not exactly the same. Sometimes we use ratios of LDL to HDL particle number, or ApoB100 to ApoA1, or triglyceride to HDL ratio. These are also useful in determining cardiovascular risk at a longitudinal level. But which is the best? Which number, if we only had one, would be the most prognostic of cardiovascular disease risk? It turns out it's LDL particle number. Let me walk you through why we think that. Shown here are two longitudinal studies where they assessed people's LDL particle number and their LDL cholesterol number, and then followed up either 5 or 10 years in the future. There's 4 different groups in both of these graphs. The groups are broken down into people with low or high LDL particle number and low or high LDL cholesterol numbers. Now, the first thing I want to draw your attention to is in both groups, the people who had the highest LDL particle numbers tended to also be the people that had the highest LDL-C numbers. But there's some individuals who have high LDL-P, but not high LDL-C. It turns out those people are at similar cardiovascular risk than the people who had high LDL-P and high LDL-C. On the contrast, both groups that had low LDL-P, independent of their LDL-C levels, were at lower cardiovascular risk. What this means is the most predictive measure is LDL particle number. But don't lose the lead here. Remember, most people who have high LDL-P also have high LDL-C. But those outlier cases where they're discordant tells us the driving factor in cardiovascular disease is LDL particle number. Therefore, interventions should target LDL particle number as the main outcome. In summary, both lifestyle and genetics play about equal roles in cardiovascular risk. This is actually promising news. Genetics is not everything. We can still reduce our cardiovascular risk through lifestyle modification. And we can use what we know about genetics to identify who to target for those lifestyle modifications. Most cardiovascular disease risk is associated with changes in lipoprotein particle numbers, specifically LDL particle number. These numbers can be useful for predicting cardiovascular disease risk, independent of genetic risk. They're also useful for us to understand how genetics affects particle numbers, and then how those particle numbers affect cardiovascular disease. Cardiovascular disease is a major cause of death in Western societies. Understanding who's at risk and who should be targeted for lifestyle intervention involves us understanding what their cardiovascular risk is. We can use both LDL particle numbers and underlying genetics, often related to LDL particle numbers, to assign whether or not somebody is high or low risk of a cardiovascular disease. This allows us to tailor interventions much more effectively than at the population level.